Dear students, Assalamu alaikum. Today we will discuss about somatic hybridization through protoplast fusion. This topic is from the course Advances in Cell and Tissue Culture Technology. I am Dr. Imran Ali, Assistant Professor, Department of Biotechnology and Genetic Engineering, Cohort University of Sciences and Technology. So, today's lecture outline is: first we will discuss about Introduction to Somatic Hybridization. We will also discuss the steps involved in somatic hybridization. We will discuss the advantages of somatic hybridization. And then finally we will discuss the limitation of somatic hybridization. So the first basic question is, what is somatic hybridization? It can be defined as the development of hybrid plant through the fusion of somatic protoplast of two different varieties or species, and the process is known as somatic hybridization. Now the term protoplast, as the whole process takes place or it starts from the protoplast. So what is protoplast? Protoplast is simply, as you can see in that figure, it's a cell except a cell wall. As we know that a plant cell wall is composed of mainly pectin and cellulose, a hard rigid layer which protects the cell. So removing that hard layer from the cell and the protoplast are released. So in simple definition, as proposed by Henston, a protoplast is an entire plant cell without its cell wall. Now, what are the main points related to somatic hybridization? The first point is, it always involves somatic cells. As the name somatic hybridization indicates, the entire process in somatic hybridization, the somatic cells plays a key role, as opposed to the normal development of hybrid in which the germ cells are involved. In somatic hybridization or in developing somatic hybrids, there is always an involvement of somatic cells. The second point is, for any somatic hybridization setup, we should have a complete or we should have a well-established tissue culture lab. So, a proper tissue culture setup is essential for somatic hybridization. In somatic hybridization, there is always equal contribution of cytoplasm. One of the important points is somatic hybridization overcomes the barrier of cross-incompatibility, or we can say sexual incompatibility. Sexual incompatibility is the inability of the species to breed and produce offsprings. Here, in somatic hybrids, we can overcome that sexual or cross-incompatibility by developing somatic hybrids which are not sexually compatible in natural conditions. In somatic hybridization, there is always an involvement of novel combination of genes from unrelated species. As we discussed in the earlier point, that somatic hybridization basically overcomes the cross-incompatibility barriers. So here you can see that it's a novel combination of unrelated species. Unrelated species in a sense that the species which cannot mate under natural conditions, they can develop somatic hybrids through the process of somatic hybridization. So that way we are expanding the gene pool by combining those unrelated species to produce a somatic hybrid. Now there are various types of somatic hybrids based on taxonomic relationship. We have interspecific hybrids. These are the hybrids between two different species of the same genus. And we have intergeneric hybrids. These are the hybrid plants of two different genera of the same family. We also have types of somatic hybrid. They are based on the contribution of chromosomes towards the hybrid. The first one is symmetric hybrids. In symmetric hybrids, the full chromosome complement of both the species is present. So, both the species are contributing equally towards the development of the somatic hybrid. In asymmetric hybrid, full chromosome complement of one species and only part of a somatic complement of another species is present. So, in asymmetric hybrid, one of the parent is contributing more compared to the second parent. We also have hybrids. Hybrids are normal protoplast of one species and cytoplast of another species. Now, let's discuss briefly about the methodology of involved in somatic hybridization, or the techniques involved in somatic hybridization. So, for any somatic hybridization experiment, the first step is to successfully isolate the protoplast, followed by fusion of the protoplast of desired species or varieties, then identification and selection of somatic hybrids, and finally culture of the hybrid cells. So, the first step, which is the isolation of protoplast. There are two methods which we use to isolate the protoplast. One is mechanical method, the other one is enzymatic method. Enzymatic method can be further categorized into two steps. One is the two-step method, the second one is one-step method. Now, in mechanical method, we start from taking the plant tissue. The cells are plasmolysed, and then we observe them under the microscope. We cut the cell wall with a knife, which can release the protoplasm or the protoplast, and finally we collect the protoplast. The techniques looks very simple, but it has some limitations. We will discuss what limitations it have. The first one is, it is used for vacuolated cells like onion bulb scale, radish and beetroot tissues. So, it means that this method is limited to the cells which are larger in size. The yield of the protoplast is relatively low. The process in itself is laborious and a tedious process. The viability of the protoplast is also greatly affected by isolating the protoplast mechanically. We also have a more improved and efficient method of protoplast isolation, and that is the enzymatic method. In enzymatic, we start with the leaf sterilization or removal of epidermis. The cells are plasmolysed as it was done in the mechanical method, but in enzymatic method, basically, as we earlier said, that the cell wall is basically composed of pectin and cellulose. So, in enzymatic method, we basically use enzyme pectinases and cellulases. In one-step method, we use these enzymes together to degrade the cell wall, which can release the protoplasm. While in the two-step method, we use these two enzymes separately. First, we use the pectinase, followed by the cellulase, which can then release the protoplasm and we can collect them. This method is more efficient and reliable. Now, after isolation, we fuse the protoplast. The protoplast fusion may be of two kinds. The first one is spontaneous fusion and the second one is induced fusion. So, the spontaneous fusion. In spontaneous fusion, the adjacent protoplast and enzyme mixture have a tendency to fuse together to form homokaryons, while in induced fusion, the freshly isolated protoplast can be induced to undergo fusion with the help of a range of fusogens. There are several chemicals which are used as a fusogens, and we also have a novel and a more improved method of electrofusion in which the electric impulses are used to fuse the protoplast. As you can see in that figure, in protoplast fusion, I will briefly discuss the whole process. We take two strands or two plants. In the first step, we isolate the protoplast, which are fused together with the help of fusogen, which can be one of these chemicals, or we can use the electrofusion or the electric impulses. After the protoplast are fused, it is the heterokaryon which we desire, and the heterokaryon is when in a single cell we have the protoplast from strain A and strain B. I will later discuss that in a culture product we have, along with heterokaryon, we have homokaryons also, which are not desirable product in somatic hybridization. So that is a brief history, and finally we can get that hybrid. Now, as previously discussed, the heterokaryons. Here you can see it is important to clarify that point about the homokaryons and the heterokaryons. In protoplast fusion or in somatic hybridization, we always take two plants which are unrelated or which cannot be crossed naturally, which are sexually incompatible. We take these strands and we isolate the protoplast. Now, it is the heterokaryon. Heterokaryon is the combination of protoplast from strain A and strain B, or plant A or plant B. In a culture medium, we can also get homokaryons when protoplasts from the same plant are combined together in a single cell. Similarly, like the plants from the other parent, the plant B, they can be combined in a single cell. These homokaryons are not desirable product in somatic hybridization. The desirable product is the combination of two different protoplasts from the sexually incompatible species in a single cell. Now it is quite challenging to select heterokaryons in a cultured from a cultured solution. So for that there are several markers which are used for the selection of the heterokaryons or the hybrid. The first one is based on biochemical method, in which the hybrids are selected on the basis of drug sensitivity and by the use of auxotropic mutants. There are some visual methods in which the visual selection is coupled with differential media growth, and then we also have some cytometric methods. So now the first method is based on the biochemical methods. The biochemical methods for selection of hybrid cells are based on the use of biochemical compounds in the medium, in the selection medium. These compounds help to sort out the hybrid and parental cells based on their differences in the expression of characters. As the first method is based on the drug sensitivity. This method is useful for the selection of hybrids of two plant species if one of them is sensitive to a drug. Here you can see the protoplast of Petunia hybrida in species A can form macroscopic callus on MS medium but are sensitive or they are inhibited by actinomycin D, a drug. On the other hand, Petunia parodi protoplast forms small colonies and it is resistant to the drug actinomycin D. When these two species are fused, the fused protoplast will produce large colonies because it will derive the characters from both the protoplast. So, it will acquire the character of large colonies from one parent and it will also acquire the character of growth on the drug actinomycin D. So, it is only the heterokaryon which will acquire the qualities of species A and species B and then it can grow on the selective medium. The homokaryons, they will not survive in the selective medium. So this is the first method which is based on the drug sensitivity. Now coming towards the auxotropic mutants. The auxotropes are mutants that cannot grow on a minimal medium and therefore the auxotrope requires specific compounds to be added to the medium. Here it is nitrate reductase deficient mutants of tobacco. You can see these are nitrate reductase deficient mutants. The parental protoplast of such species cannot grow with nitrate as a sole source of nitrogen, while the hybrids can grow. Two species of nitrate reductase deficiency are taken in that experiment or in that method. Now these two species of nitrate reductase deficiency is one due to lack of apoenzyme and the other lack of molybdenum cofactor, that is the CNX mutant. The parental protoplast of these alone cannot grow on nitrate medium while the hybrids can grow. The selection of auxotropic mutant is possible only if the hybrid cells can grow on minimal medium. Another limitation of this technique is the paucity of higher plant auxotrope. So apart from these biochemical methods, we have the visual methods. Now the visual selection of hybrid cells, although it's a tedious process, but it is more efficient. In some of the somatic hybridization experiment, chloroplast deficient albino or the non-green protoplast of one parent are fused with the green protoplast of another parent. This facilitates a visual identification of heterokaryons under light microscope due to the color differences of the two parental protoplasts. The heterokaryons are normally bigger and green in color, while the parental protoplasts are either small or colorless. Further identification of these heterokaryon has to be carried out to develop a specific hybrid plant. The more efficient one is to combine those visual selection method with differential growth media. Now, there exists certain natural differences in the sensitivity of protoplast to the nutrients of a given medium. Thus, some media can selectively support the development of hybrids, but not the parental protoplast. Here you can see a diagrammatic representation of visual selection coupled with the growth of heterokaryons on selection media. You can see that the species A, it has green protoplast. The species B, it is albino or non-green protoplast. They are cultured on a selection media. Alone the parental protoplast of species A, it will give small green cell colonies and no further growth on that particular medium. In species B, the homokaryons will give white cell colonies and white callus. But we will have a fusion product which will give us the green cell colonies and green callus. So, the somatic hybrid plants will be able to grow on that differential media. We can also identify the hybrid plants. So now we have the identification of the hybrid plants, and we can identify the hybrid plants by its morphology, the isoenzyme analysis of hybrid plants, its chromosomal constitution, and there are some molecular techniques in place for the identification of hybrid plants. We will briefly discuss. Now here it is important to mention that the identification of hybrid plants is done after the plants are cultured and they are grown, and it is only to verify, or it is only to identify that these plants are actually the hybrids. So the first one is the morphology of hybrid plants. Now the morphological features of hybrid plants, which usually are intermediate between the two parents, and they can be identified through a naked eye. For this purpose, normally the vegetative and floral characters are considered. These includes the leaf shape, leaf area, root morphology, the flower shape, its structure, size and color. For example, the somatic cells such as pomatos and tomatos, which are fused product of potato and tomato, show abnormal morphology, and it can be easily identified. Although the genetic basis of the morphological characters has not been clearly known, intermediate morphological features suggest that these traits are under the control of multiple genes. It is preferable to support hybrid morphological characters with evidence of genetic data. The second one is isoenzyme analysis of hybrid plants. The multiple forms of an enzyme catalyzing the same reaction are referred as isoenzymes. Electrophoretic patterns of isoenzymes have been widely used to verify the hybridity. Here, the somatic hybrids, they possess specific isoenzymes of certain enzyme of one or the other parent, or both the parents simultaneously. There are many enzymes possessing unique isoenzymes that can be used for the identification of somatic hybrids. For example, we have amylase, esterase, etc. If the enzyme is dimeric, a somatic hybrid usually contain an isoenzyme with an intermediate mobility properties. The isoenzymes are often variable within the same plant. Therefore, it is necessary to use the same enzyme from each parent from a specific tissue with the same age. The most straightforward is the chromosomal constitution method for the identification of the hybrid. Here, the number of chromosomes present in the hybrid cells can be directly counted. This provides information on the ploidy state of the cells. The somatic hybrids are expected to possess chromosomes that are equal to the total number of chromosomes originally present in the parental protoplast. Sometimes the hybrids are found to contain more chromosomes than the total of both the parents. So, the presence of chromosomal marker is greatly useful for the genetic analysis of the hybrid cells, and through the improved methods of karyotyping, this method can be used ideally for the identification of the hybrid plants. Apart from these methods, we have some molecular techniques, and with the recent advancement in molecular biology, it has greatly improved the understanding of genetic constitution of somatic hybrids. In molecular techniques, we have the differences in restriction pattern of chloroplast and mitochondrial DNA, we have the PCR technology, etc. Now let's move towards the advantages of somatic hybridization. Somatic hybridization can be useful in the production of novel interspecific and intergeneric hybrid. For example, pomato, it is a hybrid of potato and tomato. The transfer of some agronomic traits, for example, the disease resistance, the abiotic stress resistance, herbicide resistance, and many other quality characters. And for the production of unique hybrids of nucleus and cytoplasm, known as hybrids. Some of the limitations of somatic hybridization: its regeneration capacity is normally poor. It is a great challenge to culture the fused products because its viability is a difficult job to maintain. This method is not successful in all plant species, and we also have sometimes the production of unfavorable hybrids. Lack of an efficient method for selection of hybrids is an important limitation, although we have some methods which are used for the selection of hybrid, but still a more efficient method is required to efficiently identify the hybrids. These are the references. That's all for today's